Over to our next question, that is DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as. Now what they are asking you, they are telling you what is, for what the DNA fingerprinting is used. Okay, they are already telling you in the question. They are telling you DNA fingerprinting involves identifying differences in some specific regions. Now they are asking what that specific regions of DNA which are different, which we are using to compare the different individuals or the difference among them, okay, in the DNA fingerprinting procedure. Now let's move on to the options. First, repetitive DNA. Second, single nucleotides. Third, polymorphic DNA, and fourth is satellite DNA. So the correct answer for this question is fourth option, that is satellite DNA, because the differences in some specific, that specific regions of DNA which is different in different individuals are VNTRs, which we are using in DNA fingerprinting technology, that is variable number tandem repeats. And VNTR is the example of satellite DNA. So that's why the correct answer for this question is your fourth option. Thank you. Over to our next question, that is plasmid PBr 322 has PST1 restriction enzyme site within gene ampicillin resistance that confers ampicillin resistance. If this enzyme is used for inserting a gene for beta galactoside production and the recombinant plasmid is inserted in an E. coli strain. So what they are saying, the question is asking you, this is PBr 322. Over here they are having ampicillin resistant gene also, and a part of that they are having the site for PST1 enzyme. And what they are doing, this to this enzyme, what they are incorporating, and they are incorporating a gene that is beta galactoside. Okay, so let me clear one thing. Whenever you are inserting a different gene or an foreign gene or foreign DNA or desired DNA to a particular vector or a plasmid, then there will be there will be an inactivation of a particular gene. Okay, this is the principle I'm telling you, but the answer we can tell by looking forward to the options. Let's move on to the option. First option is saying that the transformed cell, that means after the transmission of this insertion of beta galactoside gene, that is called transformed cell, will have the ability to resist ampicillin as well as produce beta galactoside. Okay, so ability, now that will produce beta galactoside, this is correct, but it will resist ampicillin, this is not correct. So we can't go with option number 1. Second option, it will lead to lysis of host cell, no it will not. Third, it will be able to produce a novel protein with dual ability, that is also not correct, because in transformed cell that is not showing, because this is something else and not going with the question. Fourth, it will not be able to confer ampicillin resistance to the host cell. So the correct answer for this question is your fourth option. Why is it so? Because in a plasmid, when you are inserting a gene over there, so a gene, that is ampicillin resistance gene, which is present, the PST which is present. So when you are inserting a foreign gene over here, so ampicillin resistance gene will get inactive, and because of the inactivation of ampicillin resistant gene, it can't able to resist ampicillin into the host cell. So that's why the correct answer for this question is your fourth option. Thank you.
Match column first with column seconds. List first, nitrococcus. This question is related to nitrogen metabolisms. Nitrococcus bacteria, Rhizobium bacteria, Thiobacillus bacteria, and Nitrobacter bacteria related to these functions. Atmospheric nitrogen convert to ammonia, ammonium ions. Biological nitrogen fixation, Rhizobium bacteria. conversion atmospheric nitrogen to ammonium, means B is for. And ammonium's ion convert to nitrate ions and nitrate to nitrite ion. Ammonia convert to nitrate ion through the nitrococcus bacteria. Through the nitrococcus bacteria, means first is conversion of ammonia to nitrate. Second, and nitrate to nitrite nitrobacillus bacteria. Nitrobacter nitrate and thiobacillus related to denitrifications. Ions convert to nitrogen's denitrification. Match the column. Nitrococcus. Second, rhizobium, conversion of atmospheric nitrogen to ammonium ions. 4, thiobacillus denitrification. First, means correct answer is 4.
Over to our next question, and that is match list 1 with list 2. So list 1 is A, protein, B, unsaturated fatty acid, C, nucleic acid, D, polysaccharide. List 2, 1, C double bond C, second phosphodiester bonds, third glycosidic bonds, and fourth peptide bonds. By looking to these options, these information, we can get to know that we have to match the molecules which and the bonds which are present in these biomolecules. So let's see. First we are talking about protein. So we know that protein is the polymer of amino acid bonded by peptide bonds. Unsaturated fatty acids are those fatty acid where at least one double bond is present and the carbon compound, so that is C double bond C. Nucleic acid, so in nucleic acid phosphodiester bond is present, because in nucleic acid we are talking about DNA or RNA, and you have seen in DNA and RNA there is sugar phosphate backbone. So sugar phosphate, they both are bonded together by phospho with the combination of phosphate, so that is called phosphodiester bond. Okay, so for nucleic acid it will be 2. And polysaccharide, saccharide means talking about carbohydrate group. So glucose, polymer of glucose forms starch, which is a polysaccharide. So the combination or a polymer of glucose by the bond, that is glycosidic bond. So for polysaccharide, third option will be matched to the D. Let's see what it is. Let's see the options. First, for A it is 1, for B it is 4, no, for C it is 3, and for D it is 2, incorrect. Second option, for A it is 2, for B it is 1, for C it is 4, and D it is 3, it is also incorrect. Third option, for A it is 4, for B it is 3, for C it is 1, and for D it is 2, this is also incorrect. Fourth option, for A it is 4, for B it is 1, for C it is 2, and for D it is 3. So here we are, the correct match is given over here in the option number 4, and this is the correct answer for this question. Thank you.
Now student, let us come to the next question. Here the question is match list 1 with list 2. Here list 1 A as phase, list 2 1 proteins are synthesized, list 1 B G2 phase, list 2 2 inactive phase, list 1 C quiescent stage, list 2 3 interval between mitosis and initiation of DNA replication, list 1 D G1 phase, list 2 4 DNA replication. Now choose the correct answer from option given below. Here the options are option number 1 A4 B2 C3 D1, option number 2 A4 B1 C2 D3, option number 3 A2 B4 C3 D1, and option number 4 A3 B2 C1 D4. So student, here which option is the correct one? We know that student if we are talking about S phase, we know that student what will happen during the cell cycle in S phase. We know that in S phase the replication of the DNA occur in S phase, so that the amount of DNA gets doubled. So student, here A is matching, it is matching with 4. So I am writing here A is matching with 4. B, G2 phase. We know that student in G2 phase proteins are synthesized. So here in G2 phase of the cell cycle proteins are synthesized. That means B is matching, it is matching with 1. So I am writing here student 1. Quiescent stage. We know that student quiescent stage is the inactive stage. Some cells which do not divide, it will enter the stage which is called the quiescent stage, and this stage is also called G0 stage. So student, C is matching, it is matching with 2. So I am writing here C is matching, it is matching with 2. Now D, G1 phase. We know that student G1 phase should represent the interval between the mitosis and the initiation of the DNA replication. That means S phase. So student, D is matching, it is matching with 3. So here I am writing D is matching with 3. Now let us look out which option is correct in this manner, where A is 4. A is 4 in option number 1 and 2. B is 1, B is 1 in option number 2. C is 2, it is in option 2, and D is 3, it is in option number 2. So student, correct answer, it is option 2. Now student, let us come to the next question. Here the question is, which of the following statement is correct? Here the options are option number 1, fusion of protoplasm between two motile or non-motile gamete is called plasmogamy. Option number 2, organisms that depend on living plants are called saprophytes. Option number 3, some of the organisms can fix atmospheric nitrogen in specialized cells called sheet cells, and option number 4, fusion of two cells is called karyogamy. So student, here which option is the correct one, as we have to pick out the correct option. We know that student option number 1 is correct one, because if we define the term plasmogamy, we know that plasmogamy, it is the fusion of protoplasm between two motile or non-motile gametes. So it is called plasmogamy. So option number 1 is the correct one. But if we are talking why option number 2 is incorrect, we know that organisms that dependent on living plants are called saprophytes. It is not called saprophyte because we know that saprophytes are the organisms which feed on dead and decaying organic matter. So option number 2 is incorrect one. Option number 3, some of the organisms can fix atmospheric nitrogen in specialized cells called sheet cells. It is not sheet cells, it is specialized cells called heterocyst, which can fix the atmospheric nitrogen. So that's why option number 3 is also being incorrect one. Option number 4, fusion of two cells is called karyogamy. This option number 4 is also incorrect, because karyogamy is not the fusion of two cells, it is the fusion of two nuclei. It is called karyogamy. So option number 4 is also being a correct one. So here correct answer, it is option 1.
Which of the following statement is incorrect? Incorrect statement identify. This question related to the photosynthesis pigment systems. First options is stromal lamella. Stromal lamella have pigment system first only and lack NADPH2 reductase. These statements are correct statements. Grana lamella have both pigment system first and pigment system second. These statements are correct. And third options, cyclic photophosphorylation involve both pigment system first and pigment system second. These statements are wrong. Cyclic photophosphorylations involve only PS first, not a PS second. Means correct answer is third.
Over to our next question, that is, Identify the correct statement. Let's move on to the statements. First, RNA polymerase binds with rho factor to terminate the process of transcription in bacteria. This is an incorrect statement because rho factor do not found in bacteria. Second, the coding strand in a transcription unit is copied to an mRNA. This is a true statement, but before that we have to look other options. Third option, split gene arrangement is characteristic of prokaryotes. So this is an incorrect statement. Why is it so? Because split gene do not found in prokaryote, they are found in eukaryotes. Fourth option, in capping methyl guanosine triphosphate is added to the 3' end of hnRNA. hnRNA is the RNA, is the mRNA which is premature. When the mRNA is premature, that means before post-transcriptional, it is called hnRNA. So they are talking about, you can take hnRNA or you can call it mRNA, one or the same thing. So what they are saying that capping occurs at 3' end. So this is incorrect because capping actually occur at the 5' end of mRNA. So this is also incorrect. Now you can easily see that the correct statement and the correct answer is given in the option number 2. Thank you.
Now student, let us move to the next question. Here the question is, in the exponential growth equation, NT is equal to N0 ERT represent option number 1, the base of exponential logarithms, option number 2, the base of natural logarithms, option number 3, the base of geometric logarithm, and option number 4, the base of number logarithms. So student, here which option is the correct one? We know that student, if we are talking about the exponential growth equation, that is NT is equal to N0 ERT. It should represent the base of natural logarithm. It should represent the base of natural logarithm. So student, here correct answer, it is option number 2.
Over to our next question, that is, nowadays it is possible to detect the mutated gene causing cancer by allowing radioactive probe to hybridize its complementary DNA in a clone of cells followed by its detection using autoradiography because option, first, mutated gene completely and clearly appear on a photographic film. No. Second, mutated gene does not appear on a photographic film as the probe has no complementarity with it. Yes, it is true. Third, mutated gene does not appear on photographic film as the probe has complementarity with it. No, it is not, because if it is complementarity, then it will visible, and the statement is just opposite what it is saying. Fourth, mutated gene partially appears on a photographic film. No. So the correct answer for this question is your second option. Now why is it? Because what they are saying, let me clear you. For example, you are talking about the cancer cell. So we are talking about a normal cell. Let it be I am taking the brain cell. Okay. So brain cell, normal brain cell having a DNA, and the cancer cell is the mutated form of brain cell. Now what we are doing, we are taking the normal cell, and normal cell, the DNA we are taking of a normal cell, and we are having probes. Now what are actually probes? Probes are the known oligonucleotide. Oligonucleotide means a short fragment of DNA which is complementary to the DNA. So we are taking the probes, the short fragments, the sequence we know about that, and that will bind to these DNA because they are complementary to that. Okay. But cancer cell, let it be this particular DNA, and there is a change somewhere, and when we fragment this DNA, there is fragments of DNA. Okay. Now we are inserting probes in it, and when we hybridize probes in it, probes will hybridize to the known DNA sequences, complementary sequences, and the mutated part which is now changed, so for that the change is not there, so the particular mutated part or the changed DNA sequences, we will not have probe complementary to that changed DNA sequence, rather all the sequence which is known will be hybridized. So what will happen? The probe will not hybridize to the changed sequence or the mutated gene of a cancer cell, and if the probe do not hybridize with the DNA, then how you can see under a photographic film? So that's why what will happen, the mutated gene does not appear on a photographic film because the probe has no complementarity with it, because that is having a complementarity to the normal gene. So that's why the correct answer for this question is option number 2. Thank you.
Match column first with column seconds. Column first related to floral formula and column second related to plant family. First, basically identify the gynoeciums based Fabaceae monocarpellary conditions. Match the A is third. Fabaceae monocarpellary conditions. Second, then bicarpellary conditions, Solanaceae, and tricarpellary conditions, Liliaceae, and bicarpellary condition again Brassicaceae. And next things, androeciums, diadelphous conditions, specific property of Fabaceae. And flower is pentamerous flower, means A is correct match is third, and second B Solanaceae bicarpellary condition and flower is pentamerous conditions. Second means Solanaceae, and third tricarpellary conditions, trimerous flower, property of Liliaceae, and next is Brassicaceae. Match the column. First, A is third, Fabaceae. Second is fourth, and third is second, and fourth is first. Means correct answer is 4. Which of the following statement is incorrect statement? This question is related to plant physiology. Chapter is the respirations ETS systems. First options, in ETS electron transport chain, one molecules of NADH2 give rise to 2 ATP molecules and one FADH2 give rise 3 ATP molecules. Basically, single NADH2 produce 3 ATP molecules and FADH2 2 ATP molecules. This statements in reverse, NADH2 give rise 2 ATP molecules and FADH2 give rise to 3 molecules. Identify the incorrect statements. This statement is incorrect statement, but read the next statements. ATP synthesized through the complex few. This statement is right statement. Oxidation reduction reaction produce proton gradient in respiration. This statement is correct statement. During aerobic respirations, role of oxygen is the limited to the termination stage. Right. Means correct answer is First, in ETS electron transport chain, one molecules of NADH2 give rise to ATP molecules. Not at 2 ATP molecules give rise 3 ATP molecules. Means incorrect answer is first.
Now over to our next question, that is, what is the role of RNA polymerase third in the process of transcription in eukaryotes? Now they are talking about the formation of RNA from DNA. That process is called transcription, and that is performed by various type of enzyme. There are 3 type of enzyme: RNA polymerase 1, 2 and 3, and they are asking about the RNA polymerase third. Let's see. First option transcribes tRNA, 5S rRNA and snRNA. So that is correct itself. In the first option the correct answer is given, but let's see other options also before saying that this is correct. Second option transcribes precursor of mRNA. No, that is performed by RNA polymerase 2. Third, transcribes only snRNAs. No, wherever it is written only, so don't look to that option that it will be correct. Fourth, transcribes rRNAs, that is 28S, 18S and 5.8S. So the correct answer for this question is your first option, that is it transcribes tRNA, 5S rRNA and snRNA. Thank you.
Now student, let us come to the next question. Here the question is, in some members of which of the following pairs of families pollen grains retain their viability for months after release? Here the options are: option number 1 Poaceae leguminosae, option number 2 Poaceae solanaceae, option number 3 Rosaceae leguminosae, and option number 4 Poaceae rosaceae. So student, here which option is the correct one? We know that student, correct answer it is option number 3. Why we can say option number 3 is correct answer? Because here it is mentioned Poaceae. That's why option number 1, 2 and 4 is incorrect, because we know that in Rosaceae leguminosae the pollen grains retain their viability for months after release. So student, here correct answer it is option number 3.
Select the correct pair. Identify the correct pairs in these 4 pairs. First pair is in dicot leaves, vascular bundles are surrounded by large thick walls, conjugative tissue. In case of dicot leaf, not a thick cell wall surrounded by, means this statement is wrong statement. And second is cell of medullary rays that form part of cambium's ring, interpecicular cambiums. 2 types of specicular cambiums. This one intrapecicular cambiums, and this one interpecicular cambiums. Interpecicular cambiums forms in medullary rays. Means second statement is right statements, and third is loose parenchyma cells repeating in the epidermis and forming a lens-shaped opening in bark. Lens-shaped opening in bark. This is wrong statement. And fourth, large colorless empty cells in the epidermis of grass cells, subsidiary cells, not a subsidiary cells. This cells is called bulliform cells. Means 4 statement is wrong statements. Means correct pair is second. Mean correct answer is the cell of medullary rays that form part of the cambium's ring, interpecicular cambiums. Means right answer is second. Next question.